thanks Dr. Raymond Chung, who's the Director of Hepatology in the Liver Center, Vice Chair of Gastroenterology and the Kevin Polly Maroney Research Scholar at the Massachusetts General Hospital, Professor of Medicine at Harvard Medical School. Ray?
Thank you so much, Jake. And let me see if I can share my screen here. Can you all see that?
You'll just want to put it in presentation mode. Perfect.
Can you see it now?
Looks great.
Okay, great, terrific. So thank you, Jake, and thank you to the organizers for asking me to represent this group, which was a considerable and formidable group, putting together the section for the CID supplement you referenced earlier, Jake, on the challenge inoculum or selection of a challenge inoculum for an HCV CHIM trial. So just as a way by way of backdrop, it's certainly critical that we select an appropriate infectious inoculum. It's the cornerstone of a controlled human infection model. It's got to be reproducible, reliably infectious, and of course, above it all, safe. But it also needs to be relevant to human hepatitis C infection. It should, in theory, recapitulate chronicity. It should be a representative genotype. It should harbor the potential for quasi-species diversity, and of course, it should be infectious. So I'll just divide this short talk into several sections: the source of the inoculum, safety benchmarks, the characterization of that challenge inoculum, some discussion about viral genotype selection, and what an initial inoculation study to generate an acute inoculum might look like. And that will segue us nicely into the clinical trial design that George will get into in the next talk. So first, the source of the challenge inoculum. Well, there are a variety of potential sources, including, of course, the best characterized animal model, the chimpanzee, which, of course, is the only robust immune competent model for hepatitis C infection, capable of generating sequence diversity. But unfortunately, between the moratorium on chimp research and as well as safety concerns regarding potential passage of unknown pathogens, the chimpanzee is no longer a viable option as a source of an inoculum. There are, in fact, small animal models, humanized mouse models, which harbor human transplanted hepatocytes that in turn can be infected with hepatitis C and can generate infectious HCV. However, these are immunodeficient mice by design, and as with any small animal model, there could be unknown animal-derived pathogens. So this presents its own set of concerns for use in a CHIM model. Cell culture-derived virus is another potential source. The work of many on this panel have demonstrated that we can, in fact, take tissue culture adapted naturally occurring hepatitis C strains, such as the very well characterized JFH1 strain, and render it infectious in tissue culture. This is, however, a genotype 2A strain and derived from a patient with fulminant hepatitis in Japan. So there are some theoretical concerns there. Moreover, these were replicated in an adapted hepatoma cell line, and so are not compatible with GMP practices. Furthermore, there's no proof of replication in humans demonstrated for virus produced in cell culture, and is therefore not a practical choice for vaccine protection trials. So I've laid out some of the non-starters or less favorable sources. We come back, of course, to human infectious serum, which, of course, recapitulates all of the essential dimensions of human infection. It captures quasi-species diversity, and, of course, is known to be infectious. We have successfully, of course, been able to develop human-derived blood products, and this is a widely accepted practice with excellent safety profiles. So in theory, human serum may be the most attractive of the choices, for a challenge inoculum. In terms of the nature of the infection from a human source, we have two stark or largely divisible choices between acute and chronic infections. Clearly, chronic infections are much more commonly available, and acute inocula are certainly far less common. They are, as we've discussed in previous talks, accompanied by considerably higher viral loads with much higher infectivity, and acute inocula have been demonstrated to be up to 1,000-fold or even higher in terms of their ability to be infectious in either animal models or tissue culture. The paradigm strain was that, of course, that Dr. Alter initially characterized, and that's the H77 strain, the genotype 1A strain that has been the source of so much insight about hepatitis C subsequently. It was an acute inoculum at high infectivity, very well characterized, and it's still, even after all these years, ample volume in storage. But it was stored under, at the time, not in the present day, clinically approved conditions. So if one were to pursue that, it would require recharacterization, including, of course, assessment of sensitivity to current directly acting antivirals. The chronic phase infection, of course, far more representative of most transmitted infections, is very common, and we can certainly find plenty of sources of chronic infection. However, what is absolutely clear is that infectivity of chronic inocula drops considerably, and that's likely due to antibody binding and immune complex formation, so that the inoculum volume required for a chronic source would no doubt increase, and with it would go the risk of adventitious unmeasured infectious agents or contaminants. It was established, again, using the H77 model in chimpanzees, that as few as 100 immune complex free or free HCV RNAs are capable of establishing infection. Subsequent studies have used something more than that, about 3,000 RNAs in most studies that have proved over the years to be reliably infectious in both chimpanzees and humanized mouse models. So if we were to take an acute serum with harboring very high copy number, we could, in theory, yield up to thousands of infectious doses. And I wanted to just draw out that contrast, and this is a table that was generously provided by Jordan, and I've adapted it for this presentation, that if one were to take an acute versus a chronic source, and again, with the essential ask that we have 3,000 uncomplexed HCV RNA genomes, or free genomes, we'd start with an already higher viral load in the acute inoculum, 10 million, say, versus 1 million from a person with chronic HCV, translating out to about 25 million RNAs, or 2.5 million RNAs, respectively. However, if one were to look at free RNA genomes, that would largely be the case for every one of those genome copies in the acute scenario, whereas antibody binding and immune complex formation would render far fewer of the chronic samples truly free as HCV RNAs. And so one using an estimate of a 3-log reduction only yields 2,500 genome copies per milliliter, resulting then in nearly 10,000 infectious doses per milliliter in the acute inoculum, as compared to about 1 infectious dose per milliliter in a chronic inoculum, thereby increasing the plasma requirement per infectious dose dramatically, from about 0.12 microliter to just over a milliliter. And if we were to take plasmapheresis and take donors, we could get, say, for 750 milliliter plasmapheresis, over 6 million infectious doses as compared to 600 in the chronic scenario. So the acute scenario certainly appears to be far more attractive. What about safety considerations? Well, the inoculum candidate would have to conform to all transfusion and blood product safety requirements, collected an approved blood bank with approved equipment, screened by standard tests, ABO blood testing, serologic and nucleic acid testing for other bloodborne pathogens such as HIV and hepatitis B, and processed and stored under GLP and GMP conditions. The donors should meet criteria for any blood product donation, and as we've discussed earlier, should be in very good health, with absolute plans for DAA treatment after donation with clear-cut documentation of cure in those donors. We should exclude those who developed acute liver failure, fulminant hepatitis, and certainly those with more advanced forms of liver disease. We should, the attractive idea of taking someone post-organ transplant, in which even higher levels of viral RNA might be present with low concentrations of immune complexes, may be counterbalanced by the coexistence of occult pathogens in that group of patients. As far as blood types concerned, the AB blood type would be preferred insofar as it is a universal plasma donor. But if we cannot identify just such a person, again, great pains to minimize inoculum size would be important. For characterization of the challenge inoculum, the primary goal is to establish infectivity and to target 100% infection in CHIM volunteers in order to maximize power and ethically acceptable study design. We would want to start with quantification of antibody-free HCV RNA, tested in cell culture and humanized liver mouse model to establish an infectivity measured in the form of MID50s. We'd want to sequence that inoculum and be sure that there were no significant resistance-associated substitutions. Once the inoculum was identified, we could then go back and evaluate the sensitivity of that inoculum to neutralizing antibodies and antisera from previously vaccinated mice or humans. What about genotype? Well, it's ideal to have a genotype-specific challenge inoculum for each of the 6 major genotypes. This might prove impractical, at least for initial CHIM studies. However, it may be useful to have more than one. So somewhere in the middle, I think 2 genotypes may be a reasonable choice, and the 2 most perhaps attractive genotypes would be genotypes 1 and 3, which are clearly most common among those at high risk for HCV transmission, namely persons who inject drugs, responsible for nearly 90% of infections between those 2 genotypes. Even though we have heard earlier that there might be slightly diminished DAA responses among genotype 3 patients, again, we would take great pains to confirm cure of the donor with DAAs before advancing with that specific genotype 3 inoculum. And we could do this as a parallel processing exercise in which collection and preclinical characterization of multiple genotype inocula could be pursued, again, simultaneously. So how would we start? And this would be, again, a segue to the ultimate vaccine study. And we would start with a first stage study. And this would be a small scale inoculation study. This would essentially establish a dose that would lead to reproducible infection. If we were starting with an infectious serum study, it may be reasonable to start with that 3,000 RNA genome dose in the first volunteer. And if no infection were to result, we could ratchet up by increasing the dose, perhaps by a log, as a dose-finding exercise. Those who do establish infection would be followed, and again, we've had that discussion this morning, for 12 to 24 weeks, then apply curative DAAs. Samples from this initial acute inoculum study in these volunteers could be characterized further and then used subsequently as the source of challenge inoculum for the more definitive stage studies, the stage 2 and 3 studies you'll hear more about in just a moment. These participants could also be controls for observation, spontaneous clearance, and vaccination studies, again described subsequently. One consideration as an alternative to infectious serum I'd just like to present would be the notion of taking molecular clones of infectious HCV that have been established for actually a number of different genotypes and subtypes, such as those displayed here, whose course is comparable to infection with HCV-infected serum, as again has been demonstrated amply, and can be successfully passaged to other chimps and humanized mice. This may represent an even safer approach than the human source inoculum, particularly as we consider regulatory constraints. And perhaps, again, the only limitation to a clonal source of an infectious RNA in this case would be, does it recapitulate the diversity of natural infection? And I think to that, the reasonable counter would be that as we have looked at transmission of acute infection, in general, despite the quasi-species existence of the donor source, we have seen bottlenecks in transmission, with generally speaking, a single quasi-species emerging as the dominant in the infected recipient. The other challenge, I think, for an infectious RNA would be, can it be delivered without direct intrahepatic inoculation, such as was performed in the original chimpanzee studies of infectious cDNAs. So to this point, there are actually, I think, advances in therapeutic technologies that have allowed us to deliver peripherally RNAs into patients. And this has been aided and abetted by the use of cationic lipid nanoparticles, which have been well established and tolerated in clinical trials, including trials of not just vaccines, but gene therapy, RNAi and antisense approaches. And RNA lipid nanoparticles could be produced in a GLP-GMP facility for human application. And the great majority of these would naturally target after intravenous inoculation to the liver. So this may represent an attractive approach to generate challenge inocula in humans. So to close, this is what a stage 1 study might look like in order to establish that challenge inoculum using a source, again, whether an acute patient or chronic patient, or potentially even an infectious RNA tagged to a lipid nanoparticle. This would, again, corresponding perhaps to genotype 1 and genotype 3, in a patient who had been subsequently cured and documented for cure. 3,000 of these genomes would then be inoculated into the initial recipient, and we would start with 1 dose per genotype. These patients would be tracked frequently pre-viremia, and then once viremia was documented, again tracked serially until a titer developed in excess of 1 million IUs per mil, after which plasmapheresis could be performed to collect large volumes of this inoculum, which would then, of course, be a potential source for future CHIM studies, which we'll hear more about. At the point of either seroconversion, the development of severe hepatitis, or after the second plasmapheresis has been completed, these patients would then be offered curative therapy with glucapavir and probenespir, just as an example. So with that, I'll just conclude by saying that selection of a suitable challenge inoculum is absolutely cardinal for successful CHIM. It must be reproducible, reliably infectious, safe, and relevant. It would appear that human infectious serum from a high titer acute HCV infection may be the most attractive source. We should establish infectivity in animal models and then plan initial dose finding inoculation studies. The stage 1 study I just described. Infectious clonal RNA may be an even safer and more tractable alternative to generate an acute inoculum. And we should strive for several genotypes, and I would make the case that genotypes 1 and 3 may be the most attractive among those. And again, the hope here is that this ideal challenge inoculum will put us in the best possible position for success of a candidate vaccine trial. So thank you for your attention.
All right, Ray. Well, thank you very much. I certainly could not have done it better, even though you referenced my namesake as that particular CID article. So we can move on to the next presentation. Ray, maybe you can unshare. Great. All right. Well, next presentation is by Jordan Feld. I introduced him already. So without further ado, take it on, Jordan.
Jordan, I think you might be sharing the wrong screen. We're seeing the presenter notes. I think you also might be on mute. Jordan, do you want to come off mute, please?
Yeah, sorry about that. I'm just, do you have the right screen up now?
Yes, you just have to put in a slideshow.
Yeah, okay, sorry about that. I did sometimes I have two screens and sometimes it shows the wrong one. There we go. Good? Or you're still seeing the presenter?
Yep, that's good. Yep.
Okay, apologies for the delay. Thanks, Jake, for the introduction, and thanks to everybody for the participation in what's so far been a great session. And so what I'm going to talk about now is really thinking about clinical trial design, and I'm going to summarize some of the discussions that we had that we put in the CID supplement, and I've acknowledged the co-authors on that article below. So I'll talk about really the strategy here, vaccine endpoints. We've talked about some of these things already today, but I'm going to highlight where we at least felt that the consensus coming out of these workshops that we've had, as well as discussions leading up to now, point us in the direction. So the endpoints, the population, the actual trial design, what we're going to do in terms of the indications for treatment, a fair bit about safety considerations, and then some futility criteria. So when we think about this, it's important to really think about what are the preferred endpoints for any vaccine study. And it was actually discussed in the panel discussion earlier that Graham Foster said, well, of course, you're going to aim for sterilizing immunity. And I think all of us would love to see sterilizing immunity. That's the idea that you prevent initial infection. There's clear benefits to that. There's no acute infection. There's no transmission after acute infection. And the best thing about it for a CHIM is it's actually very easy to assess. The problem with it is it may be a very high bar. The alternative would be to prevent chronic infection. So in this concept, you would aim to effectively increase the spontaneous clearance rate in people who receive the vaccine. And the benefit here is, although you don't prevent acute infection, you still prevent the long-term complications of hepatitis C, particularly of advanced liver disease and of non-liver related complications and liver cancer. An alternative approach in theory for some pathogens is to try to attenuate the infection, so you have reduced severity of infection, sort of like what's been seen with COVID-19. This clearly isn't feasible in a Hep C CHIM model because the complications happen over years or decades of infection. So if we sort of think about this concept of sterilizing immunity versus prevention of chronicity, there's no question that this sterilizing immunity would be attractive. The problem with it is that it's been difficult to achieve. So in this setting, someone has protective antibodies and presumably and potentially T-cell responses. They get an infection and they never develop viremia. This has been achieved, I'll show you, with homologous challenge in chimps, where chimps have been challenged with a virus that was used to develop the vaccine, but it has not been seen with either reinfection or heterologous challenge in chimps, and I'll show you these data in a second, whereas prevention of chronicity, this idea that somebody develops an acute infection but then spontaneously clears it like we see in natural infection, but just at a more frequent incidence in people who are vaccinated. This has been achieved with heterologous challenge in chimps, and it's also been achieved with reinfection, where we know that people develop at least partial protective immunity with lower peak viremia and more rapid clearance on repeated infections. So prevention of chronicity would avoid the major consequences of Hep C, and although sterilizing immunity might be the goal, the problem is it may miss an imperfect but still effective vaccine. And I think the feeling has generally been that the bar really of setting for only sterilizing immunity might be too high a bar, and that really the primary aim of the initial vaccine trials, including CHIM, should really be prevention of chronic infection across all of the common 6 genotypes. So just to show you some of the data, these go back for a long time from Michael Houghton's team, looking at what happens when you give a chimpanzee an infection. You can see that with chronic infection, you get viremia, an ALT elevation, and then persistence of viremia. Now, when chimps were vaccinated with the same virus that was used to make the vaccine, you could see that it was possible to develop sterilizing immunity where the chimp never became viremic despite being inoculated. And the alternative here is seeing a chimp that becomes viremic. So you can see they develop positive viremia shown here, but then clear that infection. And when you look at the collection of data from Michael Houghton's team and their studies, you can see when you do a homologous challenge that if you give, in this example, 12 chimps this vaccine candidate, an E1, E2 vaccine candidate, you can see that 7 of the 12 developed acute infection, so 5 actually had prevention or sterilizing immunity, but you can see that the benefit was greater in terms of chronic infection, and this was this difference was much more dramatic when you gave a heterologous challenge. So you give a different virus, in this case, not the virus used to generate the vaccine. And then you see that all of the chimps developed acute infection, but still only a small proportion of them developed chronic infection. And when you look at this overall, you get a high clearance rate in the vaccinated chimps, that only 16% develop chronic infection compared to 63% of the unimmunized. And so this really sort of makes the case that because sterilizing immunity is achieved only with virus used for the vaccine design or homologous challenge, it's probably a pretty high bar for us in our first efforts to make a vaccine. And prevention of chronic infection is still a major benefit, and I think should be the target, at least initially. We'd certainly look for a benefit in terms of sterilizing immunity, but if it was missed, we still want to see the potential benefit of avoiding chronic infection. So then we've discussed this question of time. What defines chronic infection? And the real question here is, how long do we need to leave someone infected before rescuing them with the AA therapy to avoid missing spontaneous clearing? And here there's a balance. If we treat too early, we may abandon a good vaccine that was going to lead to clearance, but we treated the people prematurely. If we treat too late, then we expose participants to a longer duration of infection, which has potential consequences. So typically, textbooks define chronic infection as lasting longer than 6 months, but a probably better and more physiologic or biological definition of chronic infection would be the transition to immune exhaustion, after which spontaneous clearance is extremely rare. The problem is that measuring immune exhaustion in real time is very difficult. So we really do need to decide on a standard duration of infection for CHIM or vaccine trials that really balances this idea of missing late clearance against duration of infection. So we looked at this, and a big thanks to Dave Thomas, who really reached out to a lot of different cohorts to try to get some data on this. There's actually fairly little in the literature on this. And this is because the precise timing of infection is rarely known. People often have multiple exposures, and which exposure led to infection isn't often clearly known. And there are few cohorts where they've had standardized and frequent sampling of HCV RNA over time. So they've actually pulled together data from the VIP trial, the vaccine trial, as well as the BBASH cohort, a prospectively followed cohort of people with who inject drugs, and the INC3 cohort that brought together a number of cohorts of people who inject drugs that were prospectively followed for clearance. So when you start out, it looks promising. You've got over 1,000 people in these different cohorts, but once you start excluding people for whom you can't answer the question, either because of prior infection, they received treatment, they were in a vaccine study and vaccinated, they had HIV co-infection, or they didn't have frequent enough measurement. You really need at least 2 HCV RNA measurements, and most importantly, you need to know the long-term outcome of their infection. This initial number drops down to only 45 people with solid data. Of these 45, 29 went on to chronicity and 16 cleared by between a year and 18 months after infection. So let's look at what happened here. And so in green, you see the bars of measurement of HCV-R negativity, and in red that they're HCV-RNA positive. And this is looking at 3 months and at 6 months after infection. And so you can see here the clearance group and here the chronic group. And so what you see is that most people who had cleared by 3 months, they stayed negative. The problem is, so you can see that the negative predictive value for clearance is high, it's 90%, and it doesn't really get any better at 6 months, it's 92%, but the positive predictive value for chronicity, so if you're chronic at 3 months, you only have an 80% positive predictive value of going on to chronic infection. And if you look at this more specifically, that actually 44% of the people in this cohort, albeit small numbers, that were still positive at 3 months went on to clear. So The concern is that although clearly there are significant caveats to these data, they're limited numbers, but the sense is that starting treatment at 3 months will likely miss at least some spontaneous clearance episodes. The consequence of that, if a vaccine moves clearance forward, so it happens earlier, is that you'll underestimate the clearance rate and then therefore overestimate the vaccine efficacy because you may have more people clearing by 3 months. So although this is certainly not perfect data, I think this gives us some pause about this, about being confident at 3 months. Now, the flip side is that as we've commented, the CHIM may also provide really useful data by using a standardized inoculum, not having to worry about reinfection, confounding the outcomes. This actually may allow to get better predictive models over time, so that with time, we might actually be able to more reliably shorten the duration of infection, at which point we would introduce treatment. But at least on balance, these data would suggest that 6 months would be the optimal time. So let's move to the population. It's critically important to study people at risk for hepatitis C, but it's really important to think about safety first in this model. Safety to the participant and safety to the non-study population, really thinking about the transmission risk. Because of this, at least what we have proposed for initial CHIM studies is we would really focus on a very healthy population, young, with really no medical problems, no underlying liver disease, no substance use because of the risk of transmission, important notification of intimate partners and use of barrier contraception to prevent transmission within the household, and agreement to take treatment. For subsequent CHIM studies, it really is important to document both safety and effectiveness of any vaccine in the population at risk, particularly people who inject drugs. This question of reinfection is always going to be a concern. And of transmission of the CHIM inoculum. So one possibility might be to consider including people on stable opiate agonist therapy, or also just considering looking at real world effectiveness, but it will be critical to think carefully about these decisions later. We talked earlier today about participant compensation and consent, and I think it's important to distinguish this from other challenge studies because of the duration of infection we're proposing. As Jake nicely illustrated before, the Shigella inoculum was confined to hospital for 2 weeks, which I'm sure was very uncomfortable, but it's still only 2 weeks. So we've had a number of discussions about this, and there's clearly no right answers. You get different opinions from different people. We've talked to ethicists. We have generated a community advisory board to give us some insight that includes people at risk or with lived and or living experience of Hep C, and also people with experience in challenge studies. What's clear is that we need a test of comprehension for all participants. We discussed that a bit earlier. It's clear we need to compensate people for the time of study visits, so an hourly wage, and also compensates them for travel and other costs. And then what has been proposed is this concept of a weekly stipend for the duration of infection through to the end of treatment to acknowledge the quality of life effects of Hep C infection. And we would propose to use this type of model. Now, as Ray alluded to, the design would really be a multi-stage approach where the initial establishment of the inoculum, so we've talked about really generating an acute inoculum to be used for future CHIM, and how many genotypes is debatable, but probably at minimum of genotypes 1 and 3, and then maybe others. We would have these initial Sentinel cohorts that really confirm reliable infection, that you get an acute non-severe hepatitis, and you get reliable and truly universal cure with the AAs. Once we've done that, then we move on to, we really need to understand the spontaneous clearance rate for each inoculum, because that's going to be the benchmark against which the vaccines are compared. And only then do we actually propose to vaccinate people prior to infection and match them to the spontaneous clearance population in terms of these factors that are associated with spontaneous clearance. So Ray sort of showed this first establishment of the inoculum model, where you take HCV either from a chronic donor or from this RNA LNP, and you establish this in one initial recipient who then turns out to be your donor for your subsequent CHIM, for the subsequent CHIM. So this person, upon becoming highly viremic, would be plasmapheresed, and then this serves as the acute inoculum for CHIM, at which point this person would go on to receive treatment, and this would be replicated for the different genotypes being studied. The primary endpoints of this initial establishment would be to really establish the model. So you get reliable establishment of viremia. We really want to see everyone develop viremia. and have some definitions for that they do develop an acute non-fulminant hepatitis, so there are no cases of fulminant acute HCV, and that we get universal cure of infection, certainly with 2 courses, if not on the first course of DAAs. So we discussed a little bit about the acute versus chronic donor. I think Ray really went through that very carefully. And really it comes down to this issue about the inoculum size being considered. And there will be, and it will come up, I think, in the panel about other pros and cons of a chronic versus acute donor in terms of the variability or quasi-species population between the 2 scenarios. And I think that's something certainly to discuss. But one thing that's really important here is by having a standardized inoculum, it really does give us a benchmark for comparing all future CHIM studies and vaccine candidates against a really well understood virus. So, and a single donor can provide the sample for all future CHIM studies. So once we've done that, then you go on to these Sentinel cohorts. And really, as was outlined early on in the ethics discussion, we really want this first cohort to be a bit shorter, where these people get the inoculum of what we think will be infectious, 3,000 genomes. They're followed, and then they go on to treatment. This is really just making sure that everything goes smoothly. You then go to a longer Sentinel cohort, again, small numbers of participants here. If not everybody became infected, we increase the dose here to the inoculum size to ensure that everyone becomes infected. And again, we're following people until, in this case, out to 24 weeks, but we would treat them sooner, and I'll show you what I mean by this, if they develop severe hepatitis or if they or the PI feels it's required. Then finally, we move on to the spontaneous clearance cohorts, and this is really sort of the end of CHIM as just CHIM before we introduce the vaccines. Here you need a larger number of participants, and we would estimate about 24 per genotype, maybe up to 32, and I'll show you the sample size calculations. It will depend on the spontaneous clearance rate, and this population is really trying to establish what is the spontaneous clearance rate for the CHIM inoculum at 6 months, which will be the comparison for the vaccines. And then you finally move on to the stage 4, where you vaccinate people with a candidate vaccine. They will be a similar cohort size to the population in the spontaneous clearance cohort. Of course, there will have been phase 1 studies to prove the safety and immunogenicity of the vaccine prior to doing this. And then people will get the CHIM inoculum, and we will look to see whether they spontaneously clear infection more frequently than the spontaneous clearance cohort. And this will be matched on these key determinants of spontaneous clearance to the spontaneous clearance cohort. So additional endpoints for the studies, there will be many additional endpoints: virological, immunological, clinical endpoints, and importantly, ethical and qualitative studies as well in this unique model. And then, as was discussed early on, that even if these early vaccine candidates are unsuccessful, CHIM has the potential to really inform future vaccine design by really watching this in real time with detailed immunological and virological studies could inform future vaccine candidates. Now, when do we start treatment? And this is something that we have thought a lot about. And again, no right answers here. I think early on with this first stage, we want to be pretty careful. So really significant ALT elevation, or really almost any bilirubin elevation. And importantly, of course, participant request. And in this case, if the person becomes antibody positive, we would treat them. In the later stages, we would propose to actually push the bilirubin elevation to a higher level, to 100 or 6 in American units, and I'll show you why, because it's important to think about this carefully. On the one hand, we don't want to have someone go on to develop fulminant hepatitis, and jaundice, of course, is a marker of more severe hepatitis, but it's also associated with spontaneous clearance. So if we treat people at the first bump in the bilirubin, we are going to miss spontaneous clearers, and that will really underestimate the spontaneous clearance rate. So this is really a challenge of getting the balance right. We've looked carefully at the data to try and inform this. We used data from the INC3 cohort, which was a collection of multiple cohorts of people who inject drugs with acute or recent infection. And you can see that ALT elevations were pretty significant here. You can see that the IQR getting up over 1,000 in both groups. And then in a prospective study of acute hep C in over 100 patients, you can see that about half of them had bilirubin elevations that would be clinically relevant, getting up to almost 2. And the mean bilirubin in the population was pretty high, around 80 micromoles per unit or just under 5 milligrams per deciliter. Here, jaundice was clearly associated with clearance, with a marked difference in the likelihood of clearance, and also having a very high ALT above 800 was also borderline associated with clearance. Multiple other studies have looked at this. We really did pore over the literature, and there's no clear threshold identified to predict evolution to fulminant hepatitis or acute liver failure. And there are many people in the literature with these more severe acute hepatitis episodes with ALTs above 1,000, bilirubin above 100 micromoles per liter, with no cases of fulminant hepatitis or liver failure, which gives us confidence in how we came up with those numbers. Now, we have to be thoughtful about this CHIM process, and certainly if it's not working, we should stop. And we have thought about some futility criteria. If we can't establish viremia reliably, this is a problem. We don't want to expose participants if it's not working. If we have any cases of people developing acute liver failure, this has got to be a reason to stop. Treatment failure is a concern, but I think treatment failure is particularly a concern if someone doesn't respond to a second DAA course. If lots of people are failing with the first DAA course, that's a big concern, but if anyone fails a second DAA course, we need to be very thoughtful and stop. And then, of course, if we see transmission of the CHIM inoculum to non-study participants, then that's really a problem and we need to stop the study. We've thought a lot about risk mitigation strategies here with donor characteristics, so no co-infections, no recent injection drug use to avoid unrecognized pathogens, that they would be otherwise eligible as a blood donor, except for being hep C positive, and they don't have cirrhosis to exclude potentially a more virulent strain. We would do the standard pathogen panel and repeat after SVR to exclude anything that might have been in the window period. The virus would be sequenced to exclude significant resistance-associated substitutions, and importantly, the donor of this virus will be treated with the same regimen to be used in the CHIM studies and only will be used to infect a participant after confirmation of SVR. The virus will be stored under GMP conditions. We'll have serial testing to confirm stability and infectivity. And then there will be this stepwise evaluation with small Sentinel cohorts with sequential enrollment. And after this goes well, then we do need to pick up the pace and do parallel enrollment. There will be a data safety monitoring board with careful oversight. And then, as discussed previously, participants will be offered long-term follow-up. In terms of sample size, it's difficult to be certain here. The Sentinel cohorts, there's no specific sample size. These are small cohorts that just really give you a prediction of reliable infection and allow you to think about dose escalation. But the spontaneous clearance cohorts are important because we want to develop a relatively robust estimate of the clearance rate. And we do need to consider these factors that are associated with spontaneous clearance. The vaccine cohorts will really depend on the goal. If we're trying to de-risk a candidate to justify larger trials, that's going to be a pretty small trial to show us an efficacy signal. If we're looking at trying to actually get approval based on a CHIM, then of course that's going to be a larger trial. But the spontaneous clearance rate is going to greatly influence the numbers required. So if you look at this, this is a bit of a complex figure, but just to show you, this is the sample size we would require. This is the estimated clearance rate without vaccination. With vaccination, and each of these lines with the different color squares represents the clearance rate without vaccination, so the spontaneous clearance rate. And this is estimated to give you us 80% power. So if you look at a spontaneous clearance rate of 20 to 30%, which is usually what's quoted, then you need about 10 to 20 participants per inoculum to show vaccine efficacy of 60 to 70%. And I think anything less than that, probably people wouldn't feel is worth pursuing. But it becomes more difficult if you have a spontaneous clearance rate that starts to go up. If you end up above 50%, then you start to need larger cohorts. And remember, these need to be replicated potentially for each genotype to be able to show efficacy, and that's a concern. So this is something that will evolve over time, but hopefully we'll be able to use relatively small numbers to generate a robust estimate of the efficacy. So to summarize, development of a successful CHIM requires a stepwise approach to establish the inoculum and determine the spontaneous clearance rates. Clearance at 6 months balances the chance of missing late clearance with the potential risk and practicalities of longer infection. Initial CHIM studies will be performed in young, healthy populations, but it's critical that we have subsequent evaluation in at-risk populations, especially people who inject drugs. And we need to really be thoughtful about risk mitigation strategies at all stages. This is only one HCV approach. The final protocol will certainly require regulatory input and guidance, but this is sort of a consensus from the workshops of at least one strategy that we could take, and certainly looking forward to discussion and questions about this approach. Thanks so much.
All right, well, thank you, Jordan. Thanks for taking us on the journey to figure out what we need to do for a clinical trial. So let's move on to the last talk, which is probably a little more straightforward, related to how do we leverage this particular model for basic science advances. Naglaa, who is a professor of medicine at University of Montreal and University of Montreal Hospital Research Center. She will share with us her work together with a few other people about what type of basic science studies we can include in this CHIM model. Naglaa?
Thank you, Jake. I'm trying to share my screen. Okay. Is this working now?
Yes, it's fine. You just have to slideshow.
Okay. Do you see the slideshow or?
Yes.
Okay, perfect. Okay, well, thank you. So I'll just try to present a brief summary of the article that we published as part of the supplement with a lot of input from the community, but primarily work with Andrea Cox and Chris Walker. So thank them for their hard work on this. So just to remind everybody, HCV, when the virus enters the body, it initiates innate immune responses like interferon gamma, NK cells, other activities. But one of the key determining immune mediators are CD4 T cells that actually help CD8 T cells that produce antiviral cytokines and have a cytotoxic capacity to kill virally infected cells. But also CD4 T cells help B cells that produce antibodies that are able to neutralize the virus and limit the infection, and they mediate this through different types of cytokines. Also, CD8 T cells, what we know from the work of many distinguished immunologists in the field is that you need a strong polyfunctional, meaning cells that can produce multiple cytokines and have cytotoxic activity in order to control the infection. And the immune response occurs in the liver, but we can also detect it in blood. So blood mirrors some of what happens in the liver, but not all of what happens in the liver. And that's very important when we consider vaccination. So to go to the CHIM trial, so we assume that there will be two trials, or actually the first trial, which is more of a control group, which is to establish, not the one to establish the inoculum, but basically stage 2 and 3 that Jordan mentioned, that are the Sentinel cohort as well as the spontaneous clearance cohort, and that is experimental infection with no vaccination. And then the vaccination group, which is stage 4. These two groups represent a unique opportunity to understand really the early events during acute HCV infection, because when we work with humans, unlike chimpanzees, we never really know when they were infected unless you are working with people who got infected through a needle stick, but also to understand what happens post-vaccination and whether this response is protective or not. So what we would predict is that in a context of the first group, which is CHIM without vaccination, this will resemble what we would see with primary infection, where there will be high levels of viral replication. The immune response will be delayed, induced maybe around 6 to 8 weeks. Similarly, antibodies would arise around that time. So this is a relatively long time frame. In contrast, somebody who is vaccinated, we expect that this person will already have some memory T cells and antibodies that actually will act very quickly, will blunt viral vaccination. And this is similar to a situation that we see in reinfection studies. So obviously, the kinetics are not going to be the same in both cases. And this is something that we really need to consider as we look at sampling and when we should examine the immune response. So definitely CHIM with vaccination, we expect to see immune responses early on. So we need to have sampling really quite frequent during that period versus CHIM without vaccination. We would expect that this will take a lot longer, and we need to focus our sampling more around that time. And we need to look into blood, if possible, fine needle aspirates to look in the liver, and try to get large volumes of blood through leukapheresis. So these are issues to take in consideration. And I'll come back to the importance of having standardized operating procedures for sample processing and biobanking, because these are unique trials and we really need to do this well. There are many things that we can look at. We can look in the plasma for metabolomics, multi-analyte profiling. We definitely can look at antibody responses, neutralization, virus sequencing. Again, fine needle aspirates from the liver, lymph nodes can be used to characterize the immune response, or more complex omics assay like single-cell RNA-seq, ATAC-seq, or others. But of course, we cannot do all of this at once. So we decided that we need to divide this into what we would call first-line assays that are essential to assess the immunogenicity and the efficacy of the vaccine, versus second-line assays that are optional, that from which we will learn more about what happens with the immune response. So in the context of T cells, we really need to look at the frequency by looking at the cells that respond to hepatitis C antigen producing interferon gamma, IL-2, IL-21 through Ellie spots, fluorospots. We need to do proliferation assays. We need to understand the polyfunctionality of the cells, what cytokines they produce, and whenever possible, to use MHC class 1 and 2 multimers that allow us to visualize these cells and characterize them better. Second line assays would be more in-depth, high-resolution phenotypic and functional analysis. A lot of the omics assay, like I mentioned, PCR typing in order to track the expansion and contraction of particular clones. For antibodies or B cells, we definitely need to look at ELISA. We need to do ELISAs to measure antibody titers. Neutralization, for that, we need to use a standardized reference set of HCV pseudoparticles or infectious virions. And now with the developments by several groups, we can visualize B cells using E2 multimers. So we think that these are important first-line assays, while second-line assays would look more deeper into the antibody response by looking at E2 CD81 binding inhibition, repertoire analysis, isolation and characterization of the different antibodies, as well as omics on HCV-specific B cells. What is important to note is that one size does not fit all. So we need to be flexible and adaptable. We need to adapt to new findings in the field and other fields in viral, other studies in viral immunology. We need to adapt to the nature of the vaccine being tested. So if the vaccine is primarily generating antibodies, it's different from one that is targeting CD8 T cells or CD4 T cells, although we think that most vaccines will be looking at both, but the kinetics may be different. So this can be informed by the phase 1 trials of these vaccines so that we can adjust our sampling interval and what we need to look at. Finally, we need to have standard operating procedures, especially for sample collection, cryopreservation, because that will be like a huge resource for everybody. We also need to have SOPs for various protocols. It is also important to have reference samples or reference labs that would be responsible for either standardizing these protocols or providing samples that they know what the result is that can be included in the different assays. It is also important, and this is for the whole group, to think about, as this goes forward, to establish a steering committee to prioritize the study, the analysis that will be used, and also to manage sample long-term storage and access, who access what and when. So thank you, and I look forward to the panel discussion.
All right, great. Thanks a lot, Naglaa. We are running a little behind, but we should just go ahead with the panel discussion, which I'm sure will be very lively and dynamic. So Naglaa will be the moderator of this particular session, or the last session. So please proceed, Naglaa.
Okay, well, I'm lucky to have actually all three Nobel laureate on this panel, Dr. Harvey Alter, Dr. Charlie Rice, and Sir Michael Houghton. And also we're happy to have Dr. Ellie Barnes from Oxford, Dr. Julie Bruneau from University of Montreal, Jens Bukh from University of Copenhagen, and Ray Chung from MGH. And also from NIH, importantly, we have Dr. Emily Erbelding, who we hope to tackle about funding for such studies, Dr. Marian Major from FDA, and Jordan, Dave, Jake have already been introduced extensively. We also have Dr. Marc Ghany and Dr. Marc Sulkowski, prominent clinicians as part of this panel. So thank you all for joining this panel. And maybe I will start right away by actually addressing my first question to Dr. Marian. Oh, sorry.
Naglaa, you do both, Paul? Yeah.
Yes. I forgot to mention that we have the poll questions. Thank you, Jake, for reminding me. So you can see them on the screen now. So you need to answer yes, no, I don't know. The first question is, sterilizing immunity prevention of infection for HCV, a realistic goal for an HCV vaccine? The second question, if sterilizing immunity prevention of infection cannot be achieved, should a vaccine that prevents chronic HCV infection be the goal? And the third question, if prevention of chronic HCV infection is the goal, how long should we wait to treat infected CHIM volunteers with DAA? So please vote. The poll will be open during the duration of the panel, and Jake is going to announce the results afterwards.
You may have all noticed that the hosts and panelists can't vote.
Yeah, so we cannot bias the opinion. So, yeah. Okay, so we move on to the panel. So we start with Dr. Marian Major. And I think the question on everybody's mind is, what would be required by a regulatory body like the FDA to approve the CHIM trial concept for testing HCV vaccines? So I think the first part of the question is CHIM by itself. What is required? So.
Yeah, thanks for that really easy question, Naglaa. So I think actually this whole presentation today, I think, has been excellent. And what I think people have is a very good hold of what the FDA would want to see. We want to be assured of. Let me say first of all, we have had no discussions in the FDA on this. I'm not talking from an FDA guidance or thought. This is my personal perspective. But honestly, we have not discussed this within the FDA. Everything is open for discussion. And sort of argument, persuasion. So let me say that first. But I think the first thing is the safety aspect, which has been talked about a lot. It's just basically, can you infect people and cure them without any long-term safety issues? So that's the primary question. And I think that people have a very good argument for that. And I think you have the best experts to be judging that. And then the second is, what type of people would you do this in? I think initially it would have to be healthy people. And I think it'd be a small group of people. I think Jake touched on early on the sorts of things we'd want to see in terms of like toxicology, preclinical data, showing that whatever the challenge virus is susceptible to DAAs. And Ray gave a great presentation on the different options. And I think all of the questions that the FDA would have, I think are being touched on right now. And to say which one of them or which of the answers the FDA would want to have, I can't say. One thing I would say, though, is, I think that there are going to be a lot of false starts with this at various stages. And so a lot of discussion has gone into this in terms of developing the inocular, but that may end up being stalled because you might test something, you might come across something that you think is right, but it's not because it doesn't respond to DAAs in an animal model or something. So I feel as if the discussion is great, but there has to be some sort of experimental thing has to start at some point. You've got to start looking at things and making decisions and testing them in vitro or in mice, so that it helps make future decisions. There's no magic pathway that can be decided ahead of time at this point, I think. So I really think that there's a lot of really good discussion, and I think most of what the FDA would want is already being addressed.
Okay, well, that's great. So maybe Jordan Feld, you can update us. We've had some discussions with Health Canada, so if you can briefly summarize this.
Sure, I'll just very briefly. We've had 2 meetings with Health Canada, sort of with what we call a pre-CTA, which is equivalent to a pre-IND meeting, to discuss protocols, really following pretty much what I outlined in the presentation. And the first one was received with a lot of questions, and I think the second time we met with them, we really addressed a lot of their concerns, and really I think all the things that Marian pointed out about really looking at safety and really being thoughtful about the process. But they really, I think, were receptive to the concept. I think they understood the unmet medical need, and I think they want to see, as Marian outlined, that we do this in a safe and thoughtful way. But if we do, that this could progress forward, and we're proposing to hopefully think about trying to move this forward in the not too distant future.
Okay, thank you. So just to go back to the question of the population, so I think from the discussion, it would mainly start with healthy donors. PWIDs have a vested interest in that. So I don't know, perhaps Julie, Bruneau, you can comment on whether PWIDs could be included and when they should be included in such a trial.
Thank you, Naglaa. And yes, sure. I think a lot of the premises from this question has been answered today in this meeting. Jake said why it's important, and it's important to explain to the volunteers why they should participate in some kind of things like that. We also discussed the thing, and Marian just alluded to this, that from the start we have to have healthy volunteers without any type of interaction of other comorbidities or other things that unmeasured confounders, but also risk of transmission, which is very important. So what I would say is that hasn't been maybe stated is that we know that we have to involve community, and we have to involve community from the start, not from the start of when we're thinking of infecting them, but from the start of this process right now, so that they see the evolution and what we're doing and why, and they begin to think of why it's important, and why it's important for them not only to be participant in a trial like this, but eventually as a community to be ready to be vaccinated with candidate vaccine that would work, because we've seen with COVID that this community is not necessarily the one that is the most trustful in vaccines in general. So I think there is a lot of places where people who inject drugs should be involved, but they should be involved from the start. And they should see that they're not the guinea pigs. The guinea pigs are healthy volunteers and a lot of other people. And eventually, maybe one day sooner, will be integrate a part of these people that will be interested in being involved in this organization. So I think it's a step-by-step and multi-pronged approach.
Okay, thank you, Julie. So I think this is a good segue to the next question, which I will address again to Marian, and I'd like to have input from Mike and Harvey, as well as Charlie. Assuming that CHIM gets approved as a concept, what would be the essential preclinical benchmarks that the FDA would require to test a particular vaccine in a CHIM trial? Let's say we have like 3-4 candidate vaccines. What is it that you're looking for? So Marian, briefly.
I think it would it wouldn't be necessary. It wouldn't be the FDA that decides which one gets tested. It would be the whoever's sponsoring it. We'd want to see good phase 1 data to show that there's a good immune response, and maybe there may be more of a requirement to look at several of the immunological markers that you laid out, Naglaa, rather than just looking for simple immune responses at a very low level, like is it antibody positive, is it neutralizing antibody, is it T cells? Maybe we'd want to see something that's a little bit more detailed before testing that in a CHIM. That's my thought, but it would be a phase 1 trial.
Okay, thank you. So, Mike, you already have a vaccine that passed a phase 1 trial and was tested in chimps. What do you have something to add to that?
Yeah, well, as Marian said, regulators would want to see the phase 1 data. And in terms of the immunogenicity endpoints of that, you know, personally, I would recommend that the recipients are eliciting broadly cross-neutralizing antibodies against most of the major genotypes of Hep C seen around the world. I feel that those antibodies should be targeting several linear and discontinuous epitopes that are conserved among the HCV genus. You know, as an extreme example, you wouldn't want to see neutralizing antibodies and they're all targeted to the N-terminal hypervariable region of E2, right? Because obviously we know the virus can evade those very, very quickly. So, you know, Graham raised the issue earlier, you know, how cross protective do we expect a vaccine to be? You know, I guess I would say we would want to see at least partial in vitro neutralization of most of the strains of Hep C seen around the world. And yeah, in the trial that NIH sponsored many years ago, when I was in the US, where we just used E1, E2 recombinant antigen with a pretty weak adjuvant called MF59, we did publish in 2013. John Law in my lab assayed for cross-neutralizing antibodies, and we got evidence, you know, surprising evidence, to be honest, that we could neutralize at least partially most of the clades seen around the world. But I think you don't want to just look for cross-neutralization. I think you've got to understand the epitopes that those antibodies are targeting. And there are some good linear neutralizing, cross-neutralizing epitopes, and there are plenty of cross-neutralizing epitopes that are discontinuous. And, you know, some of those require both E1 and E2 to interact to create those epitopes. So, you know, that's why personally I've always favored the heterodimer of E1 and E2 as the preferred antigen. And then, you know, comments around the cellular immune responses, I would like to see in a phase 1. I'd like to see, obviously, T helper responses, as you pointed out, Naglaa. But I would like to see multiple T cells responding to the vaccine that are restricted by multiple MHC class 2 alleles, and that shouldn't be that difficult. And talking about CD8 positive cytolytic cells, I think, well, I think both for CD4s and CD8s, in my opinion, it's important not just to see those types of T cell responses, but to actually see them reacting to epitopes that are highly conserved amongst the genus, you know, because, you know, if you use a big target antigen, a large non-structural polyprotein, for example, there's a very good chance the immunogenicity will be dominated by T cells, 2 variant epitopes. and we know the virus can evade those. So I personally, planning next year, I would hope to see CD4s and CD8s to highly conserved epitopes amongst the genus. And with CD8s, which will be probably the bigger challenge than CD4s, I would want to see those CD8s, the conserved epitopes, being presented by multiple class 1, MHC class 1 alleles, and to be confident that in the population, eventually the vaccine is going to be used in, the majority of the population has the MHC background, class 1 and class 2, to present those epitopes in the majority of the population.
Thank you, Mike. Harvey, do you have anything to add?
Yeah, it's hard to add to all this information that's been out there. But so first I want to say, I mean, this has been so well thought out. Both the publications and this symposium today, workshop, whatever this really is. The presentations have been so well thought out. So we're already very advanced in this CHIM process, which I think is an essential process. I think of all the issues complex issues at every stage, choosing the inoculum is probably the most difficult. And I think it gets down to, do you want to use a patient sample, which might be ideal, acute phase patient sample, but that would have to be identified. Do you want to get that acute phase sample from an actual volunteer, infect somebody so they can be the challenge inoculum themselves? And the alternative is to put out a feeler to ASLD, EASL, all the members, and say, if you identify a case of acute hepatitis C, this is what we want you to do. We want you to get an apheresis unit. We want you to get it in this condition. We want you to store it in this way, because you only need 1, 2, 3 acute cases to have your inoculum. Had Hutchinson still be alive, he would be one of those donors. I think we need a son of Hutchinson.
Okay.
The other alternative is to use the clone virus. We have all the genotypes very well characterized, and I think Jens Bukh needs to talk about that, because this is another viable approach, and the one would have to prove that a clone virus would be equivalent to a human primary infectious inoculum.
Okay, thank you. And Charlie, would you go ahead, with a clone rather than an infectious inoculum?
Well, I mean, I think well, we know a lot about 877, so I am kind of a fan of Harvey's gold mine. And I think, the sort of lipid nanoparticle sort of inoculation strategy could, very much allow us to sort of create sort of a good challenge stock in a human volunteer. So I kind of favor, that approach, although I think, we could certainly explore many of them. And I mean, Harvey's right. I mean, I guess when these acute cases come up, that is an opportunity, but I don't know, we would still have to worry about the source. We couldn't pre-select, the person who is going to produce the challenge stock. So, other viruses, other things would have to be sort of ruled out. So I favor, maybe starting with a molecular clone. I think the issue of quasi-species complexity is something that we're not going to really be able to address, sort of in the challenge stock, because if you infect, somebody with, 3,000, sort of genome equivalents or whatever, you're not going to be capturing, sort of the complexity that there might be in, say, a chronically infected, individual. And of course, once that gets into an individual and starts amplifying, there is going to be, sort of quite a quasi-species being created in vivo. So, if you really wanted to try and cover the quasi-species landscape, then you would be going with higher inocula, rather than something that has just been shown to, sort of infect in the chimp world, sort of 100% of the animals. So I think there are a lot of variables at play here, but I do think that even just sort of choosing one genotype, one inoculum, and sort of really setting up this CHIM resource, has great value. But I think that Michael's on, the other bottleneck that we face is really sort of getting phase 1 data. Because there are so many different attractive ways of vaccinating people these days with, whether it's lipid nanoparticles or adjuvanted subunit vaccines or whatever, a lot of things that you would like to try. But you need that phase 1 data, which I'm sure sort of Mike and others can comment on this, but it's not, it still requires quite a few resources to sort of generate that database that sort of Marian and the folks at the FDA are going to want to see, and we're going to want to know, as individuals as groups trying to develop vaccines, which ones really fulfill the criteria that Mike just outlined. So anyway, I'll stop rambling on, but I mean, there are still some challenges here, but setting up the CHIM model will be helpful. But it doesn't solve all of the barriers that are there to sort of getting to the finish line.
Okay, thank you, Charlie. So there are a couple of questions on the chat with regards to your comment, Mike, about the discontinuous linear epitope. So perhaps you can answer that on the chat. But just quickly to finish with, because I'm conscious of the time, should we aiming for a broad vaccine to start with? Ellie, perhaps you can address this.
Yeah, Naglaa, you mean a vaccine that will target multiple genotypes?
Yeah.
When you say a broad vaccine?
Yeah.
Yeah, I'm open-minded about this. I think there is a place for more genotype-specific vaccines in certain parts of the world. Egypt's a pretty good example. Parts of Asia, where the genotypes are definitely more predictable. I think in the States and the UK, where we've got a mixture of genotype 1 and 3, which are genetically distinct as you get between the genotypes, we will need vaccines that target multiple genotypes. And to be honest, if a vaccine targets genotype 1 and 3, it probably will work against them all, because it will be working against the segments that are conserved between 1 and 3, and therefore between most of the genotypes. So I'm open-minded to this, but I think it would depend on the target population. And you might take different approaches for antigen design for sure, depending on your target population.
Okay. If there's anyone else would like to add, you can raise your hand, your virtual hand, before I have one final question. Okay, so Marc.
Yeah, thanks, Naglaa. I just wanted to follow up on, this is a question, again, either for Harvey or Charlie to address on this issue of the viral quasi-species, and what are the relative, benefits and downsides to, perhaps using a sample that's enriched for viral quasi-species, because I guess if you're going to use a sample from a patient with acute HCV infection, that's more likely to be a homogeneous viral species, whereas from a chronic patient, it's more likely to be enriched with quasi-species. So I'd like to just hear what are the potential, upsides and downsides to each of those approaches, and which would be preferred.
Harvey, do you want to tackle that? You already had your hand up as well.
Charlie might answer this better, but it is a balance. I think it would be ideal to have a fully developed quasi-species in a chronic patient, but that has to be balanced against the advantages of an acute phase patient that is not immunocomplexed and that reaches higher titer and is more infectious. So I think that's a consideration. And I think that has been considered, and in balance, this group has leaned towards getting an acute phase, single, more limited, more monoclonal type of inoculum. The point I was trying to make is maybe I'm missing something here, but we are not developing a vaccine. We are developing a model to test the vaccine. So I think we have to be ready, no matter what the manufacturers might come with. Will our CHIM model be able to say yes or no? This is a vaccine that should go or not go. So it's a little different perspective on what our role is compared to if we were a lab or a manufacturer actually making the vaccine.
Okay, thank you. Ellie, you have something to add, and then Jake.
Yeah, I mean, I've got 2 points. I'll try and be quick. One is that there are multiple groups now trying to make HCV vaccines across the world. And even if we establish successful CHIM models, we're trying hard in the UK, you're trying in Canada, there will be relatively limited access to those models for the number of vaccine candidates potentially coming through. And I do think we need, as a kind of group, to be having very open ways of assessing immunogenicity and comparing vaccine immunogenicity before moving on to the CHIM. And that would mean us kind of agreeing to have standardized panels, probably in an independent lab, to assess broadly neutralizing antibodies in the way that Michael described, and T-cell immunity, so that we can really compare these vaccines head to head before they move through to CHIM studies. And we should be talking about this in parallel with the CHIM, otherwise, because that's going to take some time to set up and a lot of agreements. Justin Bailey and Thomas Peachman have these standardized panels, but people will have to submit their plasma and serum for testing and be prepared to fail in that process. So that was one point. And I would also like to challenge the 3-month, 6-month endpoint that was so beautifully argued for, because I actually thought a negative predictive value of 90% and a positive predictive value of 80% at 3 months wasn't bad. And there's significant advantages to a shorter endpoint before you treat patients, including the fact you can get more patients into the CHIM, which allows for more heterogeneity in your population. Lots of other things matter, interferon lambda, gender, and you can test more vaccines, obviously, if you've got a shorter duration of infection and people are in these kind of studies for long. So I do think there's other arguments to be made about the duration of infection, which could be further debated here, because those actually predictive values were not so bad for 3 months, in my view.
Okay, I see Jordan shaking his head quickly.
Yeah, I mean, it depends a little bit on how you look at it, because it's true. It actually depends on the chronicity rate. So the positive predictive value was really because the chronicity rate was relatively high. So the positive predictive value overall of chronicity was decent, but the bigger point was that the 44% of the people who were positive at 3 months cleared. And that's not quite captured in the positive predictive value. So it's not like, it does depend a bit. And I do think it can give you a bit of a false sense of security, because it's very likely that a vaccine will shift clearance earlier, which could have more people clearing before 3 months and sort of give you a spontaneous clearance rate, which is falsely low. That's the problem. So it gives you the impression that the vaccine works better than it does.
Okay.
We can chat about it more. I mean, I agree with you. There's debate here for sure. It's not clear cut.
Yeah.
Just to be clear.
Okay. So maybe final comments from Jake and Marian, because then I have one last question for Emily. Jake, quickly.
Yeah, I just want to quickly go back to the homogeneous inoculum as an acute phase serum versus quasi-species in chronic. I think, as Ray mentioned, that if you look at those recipient of acute infections, they tend to be a bottleneck. Very few quasi-species actually transmitted or established infection in those acute infected individuals. And point number two is that I think people tend to overemphasize quasi-species. I think quasi-species is a result of chronic infections, right? It's probably had nothing to do with sort of the pathogenesis of acute infections. And then also study from like many of the CHIM studies using monoclonal inoculum, you get the same natural history of the in chimpanzee, I meant. So basically the chronicity, pathogenesis is really not that different, right? Between using sort of a homogeneous inoculum versus something have more quasi-species. So I think that's the point that I would certainly be in favor of. There's nothing wrong with using a homogeneous inoculum as either in acute phase serum or in molecular clones. So,
okay, thank you, Jake. Marian, final comment?
Yeah, I would follow on from what Jake said there, is that I do think that whatever the inoculum is that's chosen, I think a case is going to have to be made that it is representative of a natural isolate. And that can be either through sequence analysis of acute phase. I do think that some of this data needs to be generated and should be shown and used as an argument for whatever the inoculum is. But the other thing is that, I mean, are most infections from chronically infected people? So is there going to be a need to show that what you're going to be challenging with in the CHIM after vaccination is representative of what is going to be the infecting virus in just a general circulating population?
Well, I was going to chime in. Perhaps Julie can follow in terms of following these large populations. So perhaps the Hopkins people have. My understanding is that actually a lot of community-based transmission very often occur in a circle of friends, somebody who had acute hepatitis C and sharing needles, whatever, spread among their groups. So that's actually is another way of how the spread occurs. Somebody who actually had acute hepatitis C end up transmitting a huge number of their friends. So I think that's still, it's not clear whether it's really chronic infected individual has been the sole source of transmissions, right? I think that's an argument.
Yeah. Jake, based parallel to that was, if you look at sexual transmission of hepatitis C, it, I think, virtually only occurs with acute infection, or because we have followed many chronic, over 100 chronically infected people and their sexual partners with no transmission. So the chronic phase is less infectious, maybe because of complexing, maybe other factors, lower titers, but I think chronically infected people are less infectious, at least in that sexual model and probably in the IV drug use model as well.
Okay. And Julie, would you like to add something?
No, I agree. And there are some evidence from network analysis, that is the clusters of individuals that in the acute phase of the infection that drives the transmission. Right?
Okay. Thank you. Kim, do you want to add something?
We definitely have shown that acute infection is more that period that people are more likely to transmit, who are people who inject drugs, compared to chronic infection. We've shown that in our partner studies. But interestingly enough, we've also shown that these folks are they change their partners and sexual networks at a very high rate. So it's like a washing machine out there. Sometimes it gets out plenty of times. Half the people who get infected, you can't trace who they got it from because it's not one of their named partners. We've shown that too. So, but acute infection is definitely more infectious time for transmission.
Okay. Thank you, Kim. Mike, final comments?
Just wanted to mention Fred Prince's old studies. He used a chimpanzee to determine the infectivity titer of like half a dozen chronically infected patients. And he found, and this was published in a review back in the 80s. So he found the infectivity titer to be somewhere between 100 to 1,000 chimp infectious doses per mil. And of course, Harvey's acute phase H plasma is more than 1 million. It's getting on for 10 million infectious doses per mil. So in the chronic phase situation, yes, the quasi-species is much greater, but we also need to remember that most of that virus is complexed with anti-envelope antibody and is neutralized. And, that was shown very nicely by Hijikata that Jordan mentioned in his talk in Bob Purcell's lab. Harvey was a co-author on that, where the infectivity of human samples was inversely correlated to the level of immune complexes. So you can actually measure that, fairly easily in the lab, and you can figure out which samples have the most free RNA, i.e. infectious virus. You can do that for acute, and you can do it for chronics as well. So just a few comments there that hopefully were useful.
Thank you.
Can I just add to that? Because reminded me, Mr. H sample, the highly infectious H77 sample, was on the ascending limb of the ALT curve. It was relatively early in the infection. At the peak of the ALT curve, only like 2 weeks later, that material was put into a chimp and was no longer infectious. And we showed at that point that immune complexes antibody had appeared, immune complexes were present, and it was no longer infectious. So it's only one chimp, but I think that proves the point or supports the point.
Yeah.
Well, thank you very much. So I guess my final...
Naglaa, can I make just sort of one more point? And
Please,
that is,
I
think in the sort of CHIM models that have been sort of proposed, we're talking about maybe one for genotype 1 and another one for genotype 3. And I think, again, what you really want is a vaccine that is going to work in both of those models. And that would, I think, sort of filter out a lot of this concern about sort of just one inoculum being able to sort of represent, sort of all the complexity that might be out there in nature and be potentially transmissible.
Okay, very good point.
If I could just make one comment, Naglaa.
Yes, Jens.
With respect to selection of isolates, I agree with everything that's been said with acute and also with the clone. I think one advantage of picking the clone is that you can pick a sequence that's came from a patient that's well characterized, and also you can then pick something that's been developed into growing in culture, and therefore that particular sequence has already been studied against all the different antivirals that exist and that are being used in treatments. So we know inherently and not a lot about how that particular sequence will respond to antiviral treatment. And we can also pick one that's sort of representative of how it responds to antiviral treatment. We can, of course, also pick some that responds really well to antiviral treatment. You can even consider changing it so that it's very sensitive to antiviral treatment before you put it in as a CHIM. Because all sequences has sequences that give some resistance to antivirals. You can change them, and you can make a virus very sensitive to any antiviral. You can also do the opposite.
Okay,
so you get more control if you permit yourself to start that first acute infection with a clonal sequence.
Yeah.
Okay, well, a very interesting discussion. Obviously, the viral inoculum seems to be a major issue. Immunity, we have a lot to look at. Definitely, we need to have consensus on SOPs and how to move forward with that. So thank you. I think the last question, which is the elephant in the room, is about money. And I'm addressing this to Emily about potential funding mechanisms for a CHIM trial to start.
Yeah, sure. Thanks, Naglaa. So NIAID is committed to, it addresses a NIAID priority to develop a preventative HCV vaccine. We did support the VIP trial that was conducted by Andrea and Kim, and you're all familiar with that. That was more of a traditional efficacy trial, though, not a CHIM. We have an investigator-initiated clinical trial opportunity that's on our website. This would probably, what you're describing, developing the model even, might be a relatively high-priced proposal and would probably require, it would require approval by the executive committee of our institute. If it's more than $1 million direct cost in direct dollars per year, that level of approval is required. So we also have a leadership group called the Infectious Disease Clinical Research Consortium, which has their own protocol funds, and they have a lot of vaccinologists that are very experienced in clinical trials and vaccines, as well as human challenge models for a variety of things. We've really focused more on respiratory viruses, but we've done also malaria vaccine trials with malaria challenge, as well as Shigella and norovirus. So there's a lot of experience there. I think, they would have to approve use of their allocated funds in order to conduct such a study. And I think the skepticism that would come in, I think you'd need to be prepared for this, is this really less resource intensive, accelerating a timeline towards approval compared to doing a traditional efficacy trial, such as the VIP trial, which NIAID already supported. The human challenge studies that we do in flu are pretty expensive. I could probably give figures to anybody who wanted to just compare, and they're short term compared to what you're talking about with this 6-month endpoint for chronic infection. So something to think about there, whether you really would be accelerating the path towards FDA approval or any approval by any regulatory body with adding this CHIM step. I will also just add, though, that the phase 1 trial that you'd probably need that data before you went into the vaccine cohort for this. We do have resources for that. That probably would be, if you had a candidate that looked good in preclinical studies, that would be probably an easy thing to get access to our BTU sites, our IDCRC leadership group, or we have other mechanisms too that we can do those, that we can support those types of studies even without an investigator-initiated clinical trial grant. So we're committed to supporting this. I think the idea of the CHIM study accelerating progress there's probably still more discussion to have on that point.
Okay, well, thank you very much, Emily. So I think that concludes this panel. Thanks to all the participants. And with this, I hand over to Jake.